Hello, my name is Dan Szumski. I'm an independent scholar. I live in Davis, California. The title of my presentation is Introduction to Theoretical Biology. You will not be able to understand the consequences of describing the cell as a localized region of reversible process space. Because it is reversible, our cell theory is incompatible with the physical laws existing in our world. So there must be a boundary that effectively separates the physics of these two worlds and yet is compatible with both of them. We will be calling on the cell membrane to serve this purpose. An immediate consequence is the need to describe a mechanism that somehow transforms chemical constituents in the external environment into a form that is compatible with a reversible process thermodynamic structure within the cell. A similar transform is needed for substances traveling in the opposite direction. Now I want us to take another giant leap and assign different chemical rules on both sides of the cell membrane. Actually, this makes a lot of sense when you think about it. The cell interior will be characterized by redox chemistry exclusively. The exterior side will operate according to the rules of acid-base chemistry, where pH is the principal determinant of chemical speciation, and pH is defined as the negative log of hydrogen ion activity. In the cell interior, the principal biochemical determinant will be what Stum and Morgan refer to as Pe, the negative log of electron activity. In this scenario, Pe within the cell will change in a deterministic way over the cell cycle, making it the cell's dominant trajectory. Intracellular pH is not going to change very much. At the exterior of the cell, the chemistry will have a relatively constant Pe, but a highly variable pH. This is not so radical. We are merely recognizing the fact that the cell's interior is a very highly reduced state of matter. We will say that it has a high negative redox state. Here I am emphasizing the elevated electron-centered energy within the cell, and by inference, the cell's elevated covalent bond energy. Later we will discuss how and where this electron energy is localized, and you will also see how we use an obligatory electron influx across the cell membrane to drive both the continually more reduced state within the cell and also the forward progression of cell synthesis and metabolism. Now I want to do something very unusual, even by my standards. I want to change the way that we view the function of an important respiratory enzyme, carbonic anhydrase. It is known to hydrate carbon dioxide and facilitate its transport across the cell membrane. It is common to nearly all cell types, and it is one of the most common biological enzymes in nature. Its forms differ slightly across the plant and animal kingdom, but that's not important. In most animal cells, its active site has a single zinc atom with two electrostatic units of positive charge and a reduction potential of 0.68 volts. At the periphery of the active site, there are four His groups, each of which has one reactive hydrogen. Now I want you to imagine a single oxygen molecule in the aqueous environment surrounding the enzyme's active site. It has a velocity, and hence a kinetic energy of 1/2 mv squared in a deterministic sense, and an energy of kBt in a statistical mechanical sense. The electrostatic attraction of the zinc atom causes the oxygen to cleave to the enzyme's active site, forming zinc dioxide with a -2 charge. I want to clarify for you what is actually occurring. At the instant that the oxygen molecule first cleaves to the enzyme's active site, its kinetic energy drops to identically zero. It has been quieted. But the first law of thermodynamics tells us that the oxygen molecule's kinetic energy is not lost. Let me propose that this minute but calculable kinetic energy is absorbed into the enzyme oxygen complex and becomes the activation energy that the enzyme ultimately contributes to its overall chemical reaction. Think about it. We have essentially accumulated minute amounts of kinetic energy in the enzyme substrate complex into the activation energy required by the enzyme-mediated reaction. The only place where energy conversions of this kind are permitted in physics is in reversible thermodynamic reactions. In this way, we see that the two factors that contribute to overcoming the enzyme activation barrier. The first is the enzyme's information content, its very negative entropy state. And secondly, its ability to harvest waste heat from the environment. Now let's take a look at the rest of that reaction. The His groups now undergo a conformational change, a kinetic energy change where two His groups cleave the oxygen molecule, resulting in hydrogen peroxide formation at the active site. The chemical reaction is We have just stepped through a very typical enzyme-mediated chemical reaction. The energy that the enzyme contributes is derived from random molecular motion, which is temporarily stored as redox energy and then transformed back into the kinetic energy required for the conformational change of the His groups. This is the template that we will be using for all enzyme-mediated reactions in our cell model. I want you to note how this reaction fits with our theory of heat. We have systematically and very precisely transferred a very small amount of kinetic energy from the mass domain to effect a free energy change in the radiation domain. I also want you to see how at this interface between the pH and PE sides of the membrane, we are effecting a minute pH change at the exterior side of the membrane and an equally small increase in electron activity within the cell. Hydrogen peroxide is a high-energy molecule. Remember how our cell model is being built on Helmholtz free energy relationships? I want you to know that when you compare the Helmholtz and the Gibbs free energy tables, you will notice that aqueous oxygen provides an extraordinary Helmholtz free energy advantage over the Gibbs free energy. Oxygen's dissolution in water has captured free energy. Hydrogen peroxide utilizes this free energy advantage to capture the raw heat of oxygen's molecular motion for use in other transmembrane processes. We are finally ready to look at the details of transmembrane transport. The current paradigms in biology tell us that oxygen and water move across the cell membrane in passive mode. Carbon dioxide, on the other hand, is mediated by an active transport mechanism wherein the enzyme, carbonic anhydrase, accelerates its hydration to carbonic acid. This then allows the transmembrane exchange of bicarbonate ions with chloride ions in what is known as the chloride shift. I want to begin this discussion with a proposal that the more fundamental function of carbonic anhydrase might be hydrogen peroxide formation. Now, we may appear to be changing biochemistry a little too much here, but give me some latitude. Let me show you how this reaction might facilitate not only CO2 hydration, but all of the coordinated gas and ion transport across the cell membrane. I want to show you how this can be done with extraordinary precision, all the while preserving the exact stoichiometry that the cell requires. We will coordinate carbon dioxide and oxygen exchange with several known transport processes. The first will be the chloride shift, which we will also link to divalent calcium ion transport. Let me explain what I am showing here. The enzyme E is carbonic anhydrase in its protonated and unprotonated forms. It facilitates a redox reaction combining hydrogen, oxygen, and electrons to form hydrogen peroxide. It is fundamentally important that you see how electrons are captured in the hydrogen peroxide molecule and transported inward, in effect concentrating electrons within the cell. In the second reaction, carbonic anhydrase hydrates CO2, forming bicarbonate ions and molecular oxygen. The charged bicarbonate ion is expelled, while the nonpolar oxygen molecule passes into the cell interior. The chloride shift is then coupled to divalent cation transport to produce an acid-base pair and additional oxygen transport into the cell. Finally, carbonic anhydrase dissociates into its unprotonated form. We can now add these three equations, where H sub F is the Helmholtz free energy change for the reaction. The positive change in Helmholtz free energy indicates a reaction that can occur spontaneously. We will call this reaction sequence A. It links carbon dioxide excretion to oxygen uptake. I want you to take note that charged quantities will migrate to the cell exterior, while uncharged quantities will migrate inward. Now look what this has accomplished, and this is important. Two carbon dioxide molecules have been exchanged across the cell membrane for two molecules of oxygen. In this process, acid-base pairs have been formed and dissociated, at least in the thermodynamic sense. So we need to ask, do all of the intermediates actually form? The answer is yes and no. Let me explain. Ionophores in the cell membrane might merely hold the ions captive, while those continually occurring minute redox energy changes cause conformational changes to momentarily form the acid and base molecules, then in a subsequent minute energy change, acid-base neutralization happens. In this way, the ions remain where their charged forms have no adverse effects on the reversible thermodynamic process, but the required free energy exchanges still occur. Let's advance this line of reasoning with a second set of reactions that we will call reaction sequence B. It coordinates carbon dioxide and ammonia transport with sodium and chloride ions. Again, combining these three equations, but let's not stop here. Let's add one more reaction sequence to this grand canonical ensemble. You will note that reaction sequence A transports carbon, hydrogen, and oxygen across the cell membrane. Sequence B adds nitrogen transport. How about adding a third reaction sequence C that includes phosphorus transport and ATP generation? Again, combining these three equations, this third reaction sequence utilizes 231 kilocalories per mole and forms one molecule of ATP as part of the respiratory exchange of gases and ions. Ammonium carbamate, a respiratory product that expels additional quantities of carbon dioxide and ammonia, occurs in the sequence as a backup mechanism for expelling these metabolic products. Now look at what we have accomplished. In the first set of equations, we have linked the respiratory gas exchange of oxygen and carbon dioxide with a chloride shift. We have done this in an exothermal reaction sequence A, having a free energy change of plus 18.46 kilocalories per mole. It can occur spontaneously, as indicated by the positive net free energy change. However, if this were to occur, the reaction would exhibit net energy production and thus not satisfy the zero net energy change requirement of the reversible thermodynamic space. The second set of equations completes the gas exchange by including ammonia gas and adding sodium to the chloride shift. Its coupled reactions, which we have called reaction sequence B, is endothermal, requiring minus 23.54 kilocalories per mole of energy input. Finally, reaction C makes obligatory ATP formation a part of the cellular respiration at an energy cost of minus 231.49 kilocalories per mole. These last two reaction sequences cannot occur spontaneously because of their energy requirements. Now, this is a very satisfying result. However, we do need some degree of verification if we are to accept this as a plausible mechanistic framework for gas and ion transport at the cell membrane. So let's continue. We are now faced with the task of reconciling our observation that reaction sequences B and C have a net negative free energy yield and consequently are not spontaneous. This is resolved if we hypothesize a hybrid reaction combining reaction sequence A with the net negative free energy from reaction sequences B and C. This works if the hybrid reaction has a total free energy change that is exactly zero. The energy balance then becomes where A, B, and C refer to the number of reactions of each type in the overall transport process. Electroneutrality then requires that reaction sequences B and C occur in a one-to-one relationship. Thus, B is equal to C, and the solutions to these equations is. I want you to understand that this is not the final solution to this problem. In its final solution, all three coefficients will be integers. The reversible thermodynamic framework does not permit fractional reactions. The difference between 13.28 and either 13 or 14 may be due to needed changes in the reaction sequences or adjustments to the Helmholtz free energies. For example, I have lumped all of the divalent anions as calcium plus 2 and all of the monovalent ions as sodium plus 1 and calcium and chlorine minus 1. In nature, other anions and cations, for instance, potassium or magnesium, will participate in these reaction sequences. Nevertheless, we're going to accept the 14 to 1 ratio of the three reaction sequences to move forward. It is now possible to inventory the chemical composition of the three reaction sequences to estimate the total transmembrane movement of atoms. The results are illustrated here, where the first column is the number of reactions in each reaction sequence. The columns that follow are the numbers of specific atoms transported across the cell membrane by each reaction sequence. The final two columns are the number of molecules of oxygen and water transported. The respiratory quotient is calculated to be 0.94, which is reasonable. We are finally in a position to get some idea of how metabolic flexibility is achieved in the cell. Let's proceed by asking, what underlying cause-effect framework could possibly represent such a complicated transport system? And secondly, how is it modeled? Well, let's take an educated guess. Recall that we have treated all of the transport equations as a process that could proceed in either the forward or backward direction. We will begin by multiplying reaction sequence A by 14 adding one each of reaction sequence B and C to it, and viewing the overall reaction as a statistical thermodynamic process where the denominator includes the reactants and the numerator the products of those reactions. According to Helmholtz theory, the net free energy change is identically equal to zero. This expression, while it may have its inaccuracies, gives profound meaning to how the cell's metabolism adapts so easily to change. Consider, for example, a 50% decrease in oxygen availability at the cell surface. Any subset of the other factors will respond to keep the net Helmholtz free energy of the substances crossing the cell membrane at identically zero. But there is something more important here. Because this is a reversible thermodynamic system, the metabolic adjustment that actually occurs is precisely determined by the principle of least action. That might, for instance, be the adjustment that results in the smallest free energy movement amongst all of the other constituents in the equation. Do you see how the cell adapts so easily to environmental change? And also how it does so with such precision. I want you to clearly understand the versatility that this affords the cell. There are an unlimited number of altered transport combinations that can bring the net free energy back to exactly zero. But remember, from among all of these combinations, the principle of least action selects only one, and that least action selection makes every outcome precisely deterministic. Now, a scientific theory is only as good as its ability to predict outcome. So let me suggest an experiment that might be conducted to test this hypothesis. It's specialized but relatively simple. Are you familiar with the famous experiment by Sam Rubin and Martin Kamen, in which they showed that oxygen, evolved in photosynthesis, has its source in the water rather than in the carbon dioxide as was so widely believed? I propose that a similar, widely held belief impedes our ability to understand transmembrane water and oxygen movement. Did you notice that all of the intracellular water in our reaction sequences is formed at the cell membrane from hydrogen, oxygen, and electrons? No water molecules from the external environment cross the cell membrane. The precision of the cell's internal chemistry, including the number of water molecules, is very precisely regulated. So the theory is predicting that it is the active transport of water's constituent parts across the cell membrane that gives rise to biological water in animal cells. Think about it. All of the water in animal cells is made at the cell membrane. This means that intracellular water is absolutely pure and exhibits pure water's high dielectric constant. This is required by the reversible process environment. Dielectric loss of electric charge is entropy production, and it is not allowed in the reversible process space. This is the single most important characteristic of water that makes it so essential to life, its very high dielectric constant. Let me return to my description of how this hypothesis can be tested. This is an assignment for the experimentalists in my audience. You might culture E. coli in two identical experiments. The first one should contain oxygen-18 tagged water and dissolved oxygen molecular 16. The other uses the opposite recipe, having oxygen-16 water and dissolved molecular oxygen-18. We are testing the hypothesis that cellular water is formed from its constituents on the exterior side of the cell membrane. If oxygen-18 does not show up in the intracellular water in the first experiment, we have made a very fundamental discovery in biology. The same is true of the second experiment, if it can be shown that water molecules within the cell contain oxygen 18. I don't know if this is the same type of water transport that is appropriately applied to plant cells. However, the theory's reversibility constraint implies that plants also need the dielectric properties of pure intracellular water. Do you see the consequences of this? I'm proposing that some, and perhaps most of the water on Earth may have its origin in biomass. Wasn't that fascinating?